Okay. Stealth secretions. The exosomes are released and transported between neurons and glia in the nervous system, having a broad impact on nerve development, activation, and regeneration. Accumulating evidence suggests the exosomes are attributed to the pathogenesis of several neurodegenerative diseases such as prion, Alzheimer's, Parkinson's, ALS, as well as aging, in which the exosomes lack the capacity for cellular self repair and spread their enclosed pathological agents among neurons. Exosomes, gummy, blebs, tau, and your bad cholesterol. See, folks, you're not allowed to really be sick. They don't have to tell you what's inside your chronically infected Epstein borreliosis, AIDS infected cells. And this Asian scientists uncover how Epstein-Barr virus hides. Epstein-Barr virus infects more than 90% of the world's population and is linked to a number of cancers, including Burkitt's lymphoma, nasopharyngeal carcinoma, Hodgkin's lymphoma. There are currently no vaccines to prevent Epstein-Barr and other herpes virus associated cancers. They don't have to tell you how all that infectious DNA they keep shoving down your throat and jabbing in your skin kills people, like over 10,000 PTSD soldiers they murdered with psych. You're going to get it someday, but you won't be allowed to save yourself. They will tell you you're not sick because you don't have antibodies. This is cross-seeding effects of other protein aggregates. Furthermore, our nuclear magnetic resonance spectroscopic studies identified the binding spots. And our results indicate that those amyloidogenic proteins, including those contained in food and cosmetics, contribute to the aggregation, suggesting their possible role in the propagation of amyloidosis. There's little or no hope for the production of anti-trypanosomal vaccine in the near future because of antigenic variation exhibited by the parasites. Your parasites have parasites, and they have parasites. Epstein borreliosis, AIDS, infinite antigen variation you all already have. Homemade treatments. Someone asked how she's doing, Mel, and she said the homemade treatments keeps me alive. They haven't been spraying chemtrails and dumping genetically altered skeeters, bacteria and skeeters on us because they like us. The same mass murderers that gave everything real AIDS continue to do it to this day. They contaminated everything with these HeLa cells, and I linked that in one of my previous videos. On here, she does go into, there are some links here that talk about turmeric, and I'm going to do a separate video where I link to some treatments instead of this horrible news of the things that they're doing to us. Though it took 3 decades for the Geys to succeed with their efforts to create a human cell line, after their success with HeLa, culturing cells became suspiciously easy. Researchers cultivated tissue samples from their own bodies and the bodies of their families and patients. Most grew successfully. Sure, the samples struggled during the first few weeks or even months in culture, but then suddenly they flourished. Samples blossomed into full-blown healthy cell lines with the strength of, well, the HeLa cell. In 1974, a researcher by the name of Walter Nelson-Rees started what everyone called a nasty rumor. HeLa cells, he claimed, had infiltrated the world's stock of cell cultures. No one wanted to believe him. For almost 3 decades, researchers had done complex experiments on what they thought were breast cells, prostate, or placental cells, and suddenly rumor had it that they'd been working with HeLa cells all along. To believe this would believe that years of work and millions of dollars had in essence been wasted. Hmm. Uh, this link here goes into Henrietta cells in the polio vaccine, which they have tried to uh, vaccinate the world. Of course, you can't get to that link anymore. You can type in and look for that separately from the sparksoflight.org. Molecular host pathogen interaction in brucellosis. Uh, and of course, that side it says can't be reached. Brucella never went anywhere. All they did was turn off their immune systems like they did our syndromes and kids who suffer autism. Spirochetal disease seeks vectors. They knew that. This goes into after sterilizing everything and donning a surgical cap and mask, she cut the sample into 1 millimeter dice, dropped each into a tiny square into a chicken blood clot in a test tube, and covered it with a few drops of the mixture, broth, salts, umbilical cord blood that was the cell's experimental plate du jour. And then she plugged each test tube in with a rubber stopper, labeled it HeLa for the patient's first and last names, and inserted it into a crazy rig that George Gay had fashioned out of a junkyard scraps to keep the cells warm and in constant motion. A couple of days later, the little clots at the bottom of the test tubes had sprouted a whitish ring of new cells. Mary Kubicek didn't think much of it. Many of the samples she had cultured over the years had survived and reproduced initially, only to die after a few days. The next time she looked, however, she noticed that these cells weren't just growing, they were proliferating. She split them into new test tubes. They kept on multiplying, doubling approximately every 24 hours, spreading like crabgrass, as she later recalled. As long as they were kept warm and fed, HeLa cells seems to indestructible, unstoppable, potentially immortal. The Geys were ecstatic and started to send out samples to colleagues. They sent HeLa cells to Texas, New York, Amsterdam, India, Chile, in cardboard boxes packed with sawdust, the shirt pockets of pilots and air stewards, and on one occasion in the saddlebag of a mule. On the 10th of April, 1951, George Gay appeared on a local television waving a pint bottle full of HeLa and predicting the medical marvels that it would make possible. Lax has been combined with tobacco DNA to produce the first human plant hybrid. In time, however, this mid-century idealism metamorphed into hubris. In 1954, Chester Southam, the chief of virology at Sloan Kettering Institute for Cancer Research, began to inject HeLa cells into the arms of cancer patients to see if he could induce tumors. He failed to inform his research subjects that the cells were cancerous on the grounds that it might upset them, and went on to do the same experiment on healthy volunteers from a state penitentiary and on hundreds of gynecological surgery patients. In 1963, he made the mistake of trying to involve 3 doctors at the Jewish Chronic Disease Hospital in Brooklyn, whose memories of the Nuremberg doctors' trials were vivid enough for them to make the connection between its research and Nazi medical atrocities. Their protests led to others, culminating the revelation of the Tuskegee syphilis study. Let's see if that link still works. Looks like it's going to open up. My computer is going extremely slow. Dying and not dying. Kathy Gear. So I will link that article down below. It's no secret that the real Jewish people despise Zionism and what they stand for. The turd in the punch bowl. And we link back to the agent of amyloid proteinopathy in bacteria. In that instant, any tissue-specific research that used cell lines identified as HeLa contaminants was suddenly invalid. How could research on breast cancer cells be taken seriously when the cells used were actually cervical cancer cells all along. It's been estimated that over 500 research papers and more than $20 million of funding have been wasted. Their problem stems from the adolescent days of cell culture. And this is from Berkeley, The Good, the Bad, and the HeLa, which I will also link. Giving everything real AIDS was World War and nothing more. And that is literally what they have been doing. The CDC did have a video on prions, and they obviously they removed it. But they know that all these vaccines are nothing but infectious, yet they continue on. Gartler, a geneticist by training, was denounced for his lack of cell culture expertise in what must have been a profane 60s put down, was said to have dropped a turd in the punch bowl at the meeting. Predictably, his discovery was not accepted or appreciated by the cell culture community, and for many decades, his evidence was ignored. No, they did not want to admit that the HeLa cells had infected everything. Spiritual AIDS, and there's the link I shared from Vimeo and the HeLa cells. They knew in 1965 they infected everyone with infectious vaccines. You don't get to choose when yours is activated or how it behaves or is expressed in your chronically infected cells. Disturbing. It's all so very disturbing. The enemy has lied to you for decades on purpose so they could keep giving you the real cause of AIDS and cancer. They knew what exosomes was. So I will put the links to the stuff about the Geys, and my next video I will make some solutions. I'll give you some links to some things that we can do